All right, in this video we want to discuss a mechanism of horizontal gene transfer that is specifically known as transduction. So let's begin by making a frame of reference for what horizontal gene transfer is, and then we'll add some specific detail about transduction. So, gene transfer. Again, there are two forms of gene transfer. The first we refer to as vertical. This is the most common form of gene transfer in biology. And it's when an organism, like a bacteria, undergoes replication and it creates two daughter cells. So this one cell had some genes and it transferred those genes to its two offspring cells. So we just increase the amount of genetic material in the world. And then again, this parent cell transferred genetic material to its offspring cells. This mechanism of vertical gene transfer is critically important. It passes, again, genetic material from parent to offspring. This creates genetically identical offspring. Okay? This is how most bacteria are created. And again, the bacteria in the world that are created are identical to their parent bacteria. So while this is critically important, it's less interesting in that it creates more of the same kind of bacteria. What we're most interested in is how bacteria can gain traits, how they can get new genetic material. And that's where horizontal gene transfer comes in. So horizontal gene transfer is going to involve two different cells. A donor cell that's going to give away some genetic material, and that genetic material will be added to a recipient cell to create a cell that has some new genetic material. And perhaps the cell can do something new. So horizontal gene transfer passes genetic material from a donor cell to a recipient cell. Again, what's so interesting about horizontal gene transfer is that this is a mechanism by which we can create organisms with new traits. And that's critically important to understanding things like antibiotic resistance in microbial populations. So again, we want to address a specific mechanism by which horizontal gene transfer can happen. And that mechanism is transduction. Now, transduction is going to rely on the viral lytic life cycle. So I just want to remind us about the lytic life cycle for viruses. We've looked at this before. Here's a diagram. So in a lytic life cycle, a virus attaches to a cell, injects its genetic information. The cell then produces viral components for the virus. Those viral components are put together into new viral particles. And eventually that cell lyses and the new viruses are released into the environment. So we've learned about the viral lytic life cycle in the past. Again, it has these 5 steps: attachment, where the virus sticks to the host cell; entry, where the genetic material is introduced to the inside of the host cell; synthesis, when viral pieces are manufactured by the host cell; assembly, when the viral particles are put together inside the host cell; and finally release, when the new viruses are released into the environment. In order to understand transduction, we have to understand this lytic life cycle. So let's look more closely and think about what happens in this lytic life cycle that allows for transduction to occur. So we're going to talk about transduction. And quite simply, transduction is when a virus, like the one we were just talking about in the lytic life cycle, transfers non-viral DNA from a donor cell to a recipient cell. So I've got that exact same lytic life cycle diagram here, but I've modified it ever so slightly. So we've got our virus attaching to and then entering the cell. We've got the host cell manufacturing viral pieces, synthesis. Then we've got assembly, when the viruses are getting put together. And this is where I've made a slight change. I've indicated in this virus right here that the genetic material in there is different. The genetic material in this virus is meant to represent bacterial DNA, non-viral DNA. So again, transduction is when a virus transfers non-viral DNA from a donor cell to a recipient cell. This diagram is representing the donor cell. This one virus got some bacterial DNA in it, and after it is fully assembled and released, this virus containing the bacterial DNA can attach to and enter a recipient cell, but it puts in viral DNA, it delivers to the cell bacterial DNA, not viral DNA. So we just, in this transduction mechanism, moved some bacterial DNA from this donor cell that was killed by this virus to this recipient cell that is receiving bacterial DNA that was delivered by this virus. So there are a couple details in the lytic life cycle for viruses that can explain how this occurs. So let's think a little bit more deeply about a couple steps, and then we'll return to this exact same diagram. So the first thing I want to highlight is what happens during synthesis. So during the synthesis step, the very thing the virus does after synthesis begins is that it destroys the host genome. So in this cell where the virus is undergoing synthesis, there is no longer a bacterial chromosome. That bacterial chromosome has been chopped into little pieces. This is something the virus does so that the bacteria cannot follow the instructions in its own DNA and must follow the instructions in the viral genetic material instead. So in the process of sort of hijacking the host cell, the virus cuts the host DNA into small pieces. Those pieces of host DNA remain floating around in the cytoplasm. They're just not used appropriately because they're not in that chromosome form anymore. So that's one detail, all right? During synthesis, the virus destroyed the host DNA. It's floating around in the cytoplasm in small little pieces. The next piece of information that helps us understand transduction is what happens during assembly. So remember, assembly is when viral components are put together into new viral particles. And we can see that happening in this picture here. On the inside of this bacterial cell that has been infected by a virus, there are all these viral particles being put together. Okay. The problem is, when the viral particle is put together, it is possible for the viral particle to accidentally incorporate some bacterial DNA instead of, or in addition to, the viral genetic material. So mistakes happen during assembly, and some viruses contain bacterial DNA. Again, there's bacterial DNA floating around in the cytoplasm because it was cut into small pieces during synthesis. So if we look at our diagram, right, during assembly, these pieces are starting to get put together. The capsid should contain this piece of viral genetic material. But this capsid here, instead of containing viral genetic material, contains bacterial genetic material. And that explains how we can get a virus that contains bacterial DNA instead of, again, or in addition to, viral genetic material. So if we return to our diagram, again, any form of horizontal gene transfer always has a donor cell. In this case, the donor cell is this cell that is being killed by this virus when it reproduces in it. The recipient cell is this cell over here that is receiving some bacterial DNA in this viral particle. That viral particle was produced in the donor cell. And again, it contains bacterial DNA instead of viral DNA. That can happen because back in synthesis, the host chromosome was cut into small pieces, and then those small pieces can be incorporated into a virus during assembly. That virus containing bacterial DNA can drop the DNA off in a new recipient cell. What happens next depends on the function of this DNA and whether the virus did or did not contain any viral genetic material. So it's hard, you can't say for sure what happens next. The outcomes are varied. But again, all transduction is that a virus carries a piece of DNA from a donor cell to a recipient cell. So let's just point out one last detail about transduction. And the thing I wanted to highlight is that during transduction, the donor and the recipient cells, they can be separated by significant time and or distance And this is because the donor cell is dead. So the two cells are not required to be alive in the same place at the same time. Furthermore, viruses are very stable, right? We know that a viral particle is really simple, and they can float around in the environment for very long periods of time. That is even true when they contain the wrong genetic material. So one might ask, well, this virus contains bacterial DNA, not the viral genetic material. So doesn't the virus need its genetic material to do something? And I would remind students that outside of host cells, viral particles cannot do anything. All a viral particle is a sort of a container for genetic material, and that container has spike proteins on it that will allow it to attach to and enter a specific host cell. But other than that, viruses are doing nothing when they're outside of host cells. So it doesn't matter what genetic material is inside a virus. When that virus is outside a host cell, it's never using that genetic material for anything. So just keep that in mind. So we'll stop there. As always, I hope you'll let me know if you have any questions related to this content, and I will talk to you all again very soon.